Assalamualaikum everyone, welcome back to Dr. Hafsa's Obstetrics and Gynecology study channel. Yesterday we did pap smear and today we are going to do liquid based cytology, which is also called LBC in short words. So what is liquid based cytology? Liquid based cytology, it is an advanced cytological technique used for cervical cancer screening in which exfoliative cervical cells are collected and suspended in a preservative fluid. So what is the difference between pap smear and a liquid based cytology? Pap smear is just you can say a crude method for detection of cervical cancer cells, and it is a more refined sort of test in which there is a jar which has a liquid with preservative. We take the sample with a specialized cyto brush and we put this sample into this liquid having preservative. So what is the fundamental principle of this very test? It involves cellular preservation, filtration and preparation of thin uniform monolayer of cells on a glass slide, and it minimizes the obscuring factors such as blood, mucus and inflammatory exudate. So as you know that during this pap smear testing, when we put the scrapings from the cervix on the glass slide directly, then it includes that blood and mucus along with it. But in this liquid based cytology, it is an advantage that we put the sample and scrapings into the liquid, so it removes the mucus and blood and that extra debris from the scrapings. Then comes what is the methodology of this very test. We are going to talk about methodology, how we do it. The procedure begins with the sampling of the transformation zone using a cervical brush or a broom device. A broom sort of device is given usually along with that liquid containing jar. So instead of direct smearing, the device is rinsed. The device means that is that very cyto brush or the broom device with which we take scrapings from the cervical smear. This device is put into the preservative solution in the jar, and in the laboratory this sample undergoes processing, and this processing involves two kinds of methods. Either it is done with thin preparation method or it is done with short part method. So this results in standardized and reproducible cellular preparation. Then comes cytological evaluation. How do you evaluate this very sample after making the slides in the laboratory? It involves Bethesda system. So I tell you people that yesterday as we did pap smear and pap smear classification, that was old classification. This Bethesda system classification can be done on the cervical cells and the sample we take from the cervix. Either after we can do this Bethesda system classification both after pap smear and liquid based cytology. And what does it involve? It has 4 grades. Number one is negative for intraepithelial lesion or malignancy, that is NILM. Then in number 2 it is ASCUS. ASCUS means atypical squamous cells of undetermined significance, or atypical squamous cells in which high-grade lesion cannot be excluded. Then on number 3 comes low-grade squamous intraepithelial lesion, and then high-grade squamous intraepithelial lesion, and in the 4th stage comes squamous cell carcinoma. So what are the advantages of this liquid-based cytology? It improves sample adequacy, it reduces unsatisfactory smear, it has higher enhanced diagnostic accuracy, and a very important advantage of this liquid based cytology is that we are able to detect human papillomavirus DNA from the same sample, which we cannot do in the sample of pap smear. And what are the limitations of this liquid based cytology test? It has higher cost as compared to the conventional pap smear, and it requires more specialized laboratory equipment. Then what is the clinical significance? It has significantly improved the sensitivity and specificity of cervical cancer screening program, and it plays a central role in the early detection of precancerous lesion. Then integration with the human papillomavirus co-testing strategies is possible, and it has also reduced cervical cancer morbidity and mortality. Then I come on the conclusion. Liquid based cytology represents a major advancement in the cervical screening. It offers better sample quality and reproducibility and better diagnostic yield. So guys, stay connected for upcoming lectures.